Good afternoon, everyone. Thanks for the invitation, Dr. Xu and Dr. Wang. It is my great pleasure to be here and share the topic on the application of artificial intelligence and machine learning in the drug development and precision medicine. So before I start, I would like to make a disclaimer that the review expressed are those of the authors and do not reflect official policy in FDA. So here is a outline of my today's topic. So first, I will talk about the AI machine learning landscape. Basically, it's a train analysis for submissions in the FDA. So to illustrate the practical application of AI machine learning in drug development, so I will discuss three particular examples. So the first one is an anakinra review, which showcases how AI machine learning technique can be used to determine FDA's regulatory decision. The second case is machine learning-based enrichment strategy. So I will talk about the, I believe is the low-hanging fruit application for AI machine learning in drug development later. The last one is it's a review related to covariate adjustment during the clinical trial. Lastly, I will talk about some of the regulatory research activities in our office. The first one is we use a scoring rule to predict the cancer patient's prognosis. The second one is the enrichment strategy for schizophrenia trial. Today we have a lot of topics. So always ask questions if you have any. So I guess everyone here, we either stats or mathematics or AI machine learning, we know this trend, right? Why we talk about AI machine learning today? I think a lot of the reason is it provides a new tool, also can leverage large amount of data to improve the drug development. I think that's why everyone is so hyped about the AI machine learning. So from the FDA's perspective, or our team, or my personal, the most important question is, in terms of AI machine learning in drug development, what is the considerations we have? So I think that's very important for us to understand. Then the pharmaceutical company, they know what's our consideration. Then they can design so they have the strategy to address those considerations. I think that's the most important part. So last year, FDA released the AI machine learning drug development discussion paper. So this, you see the highlighted part, a landscape analysis conducted by myself was highlighted in the FDA discussion paper. So this research has played a substantial role in augmenting FDA's understanding and experience with AI machine learning in drug development. So I will talk about the paper later.
So I have a general question. So you have AI/ML.
Yeah.
So does that mean at FDA, when you talk about artificial intelligence, what do you refer to machine learning?
Yes, basically it's interchangeable. So basically, also my title is AI machine learning reviewer. So basically, it's pretty much interchangeable. Yeah. So we published the landscape paper 2 years ago, and we in this particular slides we provided updates for 2022, and also have results for 2023. We actually have more cases. So I want to clarify the AI machine learning related case only focus on the summation in CEDAR. Maybe some of you guys know in FDA there is a larger center called CDR-H. Their main focus is on the device, so they have much more prove the case, AI machine learning cases device. Let's say some of the diagnostic tool for certain disease, right? There are a lot of the algorithm is supported by the AI machine learning. Okay, so you can see this trend is very exciting, especially you can see before 2021 there are very few cases, and from the 2021 2022 we can see it's more like a 10 times difference, and this year we actually have more, but the manuscript is still, we still prepare the manuscript, but the number is more than 2022.
What is the total number?
Oh, sorry, you mean?
I mean, each year.
Yeah, this is a total number for each year.
I mean, the denominator.
We don't
in
have
total.
Oh, that okay. That's a good
Proportion in terms of
okay, yeah, that's a good we didn't count that one.
okay.
We just narrow sort of search our internal database and try to find some of the relevant summations.
So okay, we assume denominator pretty much constant.
Yeah. Okay, so you can see this is if we break down by the development stage, we can see a lot of the AI machine learning summations is in the clinical development. But I want to make a clarify. This one is pretty much underestimated the trend, because a lot of the study in the drug development stage is not here. You know, the AlphaTwo, right? Also, a lot of companies, they focus on the de novo sort of drug discovery. They try to synthesize new proteins. Those type of application is not covered here. So this one is only, I think is I think most of the summation in FDA's database is in the clinical development stage, because that's why the sponsor submit their package, right? Then we see those data. So, but in the drug discovery stage, we don't have that information. Also,
drug, no device.
Oh no, this is a drug development.
Okay, you don't look at the device.
Device is for the CDRH.
Okay.
They have much more. They have, I think they have 700 approved case. That's a lot. And I think this one is the most interesting part, oncology, right? So if we break down by the therapeutic area, we can see oncology, psychiatry are among the most like enriched AI machine learning summations. So oncology, we do have a lot of machine learning, like exploratory analysis. They try a lot of things. So also we summarize the common analysis type of AI machine learning applications. So for example, the outcome prediction, covariate selection, pharmacometrics modeling, anomaly detections, and also summarize the objective AI machine learning. So I think one thing I mentioned is, I believe the low-hanging fruit of this AI machine learning application is enrichment trial design. So generally speaking, there are 3 categories for the enrichment strategy. The first one is usually we want to reduce the homogeneity, which is we want to reduce the variability of the subjects, right? If the subjects have a very diverse sort of, we can see its homogeneity, it will sort of dilute the treatment signal. So in that case, the trial might fail. So in that case, the first strategy is we want to decrease the variability of the population. We do have an example later. The second case,
So it's some kind of subgroup analysis.
It's a pre-specified subgroup.
Pre-specified.
Yes.
Subgroup.
So you cannot do post hoc, right?
All right.
So in your study stage or protocol stage, you want to specify, say, okay, we want to enroll this type of patient.
Right.
We believe they are very
Yeah, I'm trying to understand that when you say drug development because of this whole process, I would like to know what stage, when you talk about the patient selection, I
Yes.
guess they already determine the compound. So, and that is to determine maybe it is a phase 1 trial or it's in what kind of,
Yeah, that's a
Nice.
really good question. So when I mentioned that during the drug development, I'm referring to the clinical stage. So
Okay.
basically the compound is determined. And we also know the drug should have certain effect. But we are not sure about the population, right? So sometimes if you enroll a wrong population, right, your p-value or it's larger than 0.05, right? So that means a lot of the effort is working on, is there any prior information or is there any algorithm we can rely on to select the right population to demonstrate the treatment effect, right?
so they use human to do.
You can use you can use old trials, right? You do have a lot of old trials, and you can use real world evidence, right? Because at this stage, it's still a hypothesis. You still need to do randomized trial to demonstrate your hypothesis, right?
That is the latest stage, so that's why I'm confused then. So if the new compound usually is not included then.
Oh, the population selection we're talking about here usually is phase 2 or phase 3.
Okay.
Yeah, usually when you do phase 1, if it's oncology, it's not the healthy population, right? But if it's other type, usually you involve healthy population. That's fine because you want to test the maximum tolerated dose, right? Sometimes, but to the phase 2 and phase 3, you want to demonstrate the efficacy of the treatment, right? You need to pick up some right population, right? You can either based on your prior knowledge or some old clinical trial or some like real world evidence to derive certain hypotheses. Then you conduct trials to validate, right?
So my understanding is you are talking about this phase 2 and phase 3
Yes.
trial, they use AI?
Yes,
Okay.
that's the interesting part. Yeah.
So, I kind of better understand. So the patient enrichment design basically try to use AI and advanced methods to do a better job in, doing patient subgroup selection, maybe during the trial, using information, based on historical data. Because usually, I mean, the simplest case will be just doing like logistic regression or some very simple model
Yes.
to the process. But now the innovation or the point is how to use more advanced methods.
Yes, exactly. So I want to clarify the enrichment strategy is not a new idea. Has there been a long time, right? So for example, in the cardiovascular disease area, right? Sometimes we want to enroll patient with very severe like status, right? Otherwise, if you enroll, because usually the stroke or cardiovascular disease, they have such a long like follow-up time, right? So the event is so rare. So if you focus on the mild patient, your sample size will be huge, right? So if you focus on severe patient, you might have a reduced sample size and a short follow-up, shorter trial time, right? So that's actually enrichment, right? But now we sort of apply this sort of philosophy to, we combine AI machine learning because we know it's data-driven. Maybe we can uncover something interesting, right? So that's the big picture here. It's not a novel idea, but now we can combine the data-driven approach, right? Okay, I guess now we can move to some practical sort of applications. So those are real. So those are real example. So it's some from us, some from a sponsor. So now let's switch gear to the 3 practical applications. So the first one is anakinra review. So this one is really sort of the first case we use AI machine learning to determine a suitable population, and also another importance is the product of this AI machine learning approach is incorporated into the fact sheet. So I don't know if you guys know the sort of the drug label or the fact sheet. So we basically the product of the machine learning algorithm is incorporated into the fact sheet. So we will see this later. So the goal is try to provide a criteria to help the healthcare provider to determine the treatment eligibility. So that's a goal for that. So maybe I will give some background of this anakinra. So this is a COVID drug. So the anakinra is the IL-1 receptor antagonist. So for this drug actually approved for other disease. A lot of this one is sort of related to autoimmune disease, something like that. And the company used a repurposing strategy to demonstrate this treatment could be used to treat patients with COVID pneumonia who are at risk of developing severe respiratory failure. So if you think about the mechanism of action of this sort of suppress the inflammation, right? So patient with severe pneumonia, they belongs to this category, right? So in this trial, this treatment demonstrated a very great sort of treatment effect as compared to the standard of care. But one of the issue here is the trial was conducted in Europe, and one of the very important biomarker or the inclusion criteria called SUPA. That means all of the patients enrolled in the study has a certain biomarker larger or equal to 6. And also this assay, the SUPA is a sort of information biomarker. This assay was not approved by that time, and also now still today is still not approved. So in that case, even if we approve this treatment, we don't have a way to distinguish which patient should take this treatment because we don't have this assay in the United States. And also this is COVID, and is, you know, several years ago, we need to have some like a certain way for this life-threatening disease, right? So under that situation, we in our review team try to sort of play with the tools we have. We come up with a research question: is there an alternative way to identify patients that are aligned with patients enrolled in the study, which the super high? Because we know the SUPA is not commercially available in the United States. So can we use other common lab values or baseline characteristic to surrogate the SUPA? That's our strategy. It's very easy, like right, very intuitively thinking. So we use a machine learning algorithm to develop easy to interpret, implement scoring system to identify patients with this SUPA larger or equal to 6, based on common measure patient characteristic. So by that time we have two. Oh, that's weird. So by that time we have two different, two independent study. We use a SAVE trial, the largest study we have to develop the scoring rule. We use a different study, a SAVE trial, as an external validation to validate our the developed score rule. And also we do have the post authorization study. Basically there is sort of additional study to further validate the scoring rule we developed. So this is our sort of consideration. When we talk about a machine learning algorithm in clinical study, right, we want to sort of have certain priority, right? In this case, our priority is the treatment. The anakinra was conducted in the patient with SUPA larger or equal to 6, and among those population, that population, anakinra demonstrated great treatment effect. Now, if we turn that into a sort of mathematical thinking or statistical thinking, is we want to prioritize PPV, right? Why? So here, so high passive predicted value ensure that patients identified by the screen rule are more closely aligned with those in the same more study. So if we have questions here, you can sort of, we can post here and have a discussion, because this is a very important part. The read, it's weird. So, and also we know the PPV is a prevalence dependent, right? Prevalence independent, right? Dependent, yes. That's why we have a different one, the specificity. We also want to check the specificity of this screen rule. And also, this is our sort of conservation or the priority. Then we look at the sensitivity, because we know during that time we have other type of treatment, the IL-6. So even if the patient is eligible for anakinra, but the scoring rule didn't select the patient still have other options. But we want to make sure the patient selected by the scoring rule can really receive the treatment benefit from anakinra. So that's why the consideration is a little bit, how to say, towards, sort of prioritize towards the PPV and specificity. So based on that consideration, we design algorithm with, so we actually have a let's see, what is the multi objective. So the loss function is like a constraint, right? We sort of prioritize on PPV, which means the PPV has to be larger than 0.95. In that, under that situation, we want to maximize sensitivity. That's how we design the loss function. And we can see in the training data set and the external validation data set, the performance is very similar. I think in the next slide, you will see some other consideration in the screen rule. So this is the screen rule we have. And you can see the highlighted part. Patients meet at least 3 of the following 8 criteria are considered likely to have the super large or equal to 6 at a baseline. So we turn a very complicated, potentially maybe XG boost or neural network, that complexity into a screen rule, because we know this is very easy to implement, right? And if you happen to have domain knowledge, you know a lot of the features selected have the literature support. For example, the neutrophil to lymphocyte ratio, it's an immune biomarker, inflammation biomarker. So, and also if you happen, so this is the drug label for anakinra. We put this result in the section 1.1, which is a very important part of the drug label. So we have this scoring rule to guide healthcare provider to determine the treatment eligibility. Of course, a lot of times when we talk about AI usage in the clinical setting, it's more like the human in loop. So this provides an initial sort of assessment. So the physician or the doctor will have their final decision based on this piece of information. But still, we believe this will provide a lot of information for the physicians.
So manual for these multiple objectives.
Yeah.
So you have multiple criteria, right?
Yes.
So when you, for example, when you design the loss function,
Yeah.
How to determine the importance of each part? Will be just like simple average of them, or you actually have different weights?
Oh, okay, sure. Yeah.
Criteria?
So that's a very interesting question. So in this case, we actually design a algorithm, end-to-end network method. We can sort of there are 3 parts. One part could sort of learn what is the important feature. Second component is we want to learn the cutoff for the important feature. The third part is given the cutoff for that feature, what is the weight? We designed the integer weight, 0, 1, 2, 3, or -1, -2. So it's an end-to-end learning strategy. Try to combine everything. So those one is based on the sort of end-to-end training.
So what is the role of machine learning?
Oh,
Yeah.
is try to the machine learning is so this is a product of machine learning.
Yeah. Probably this is a result
Yes.
already.
Yeah.
So to get this result, you use machine learning
Yes.
technique
Yes.
to end up with, okay.
Yes.
I
To
see.
deliver. So you can think of a logistic regression, right? Logistic
Right.
regression is more a little bit complicated,
Mhm.
but this one is more straightforward. You just
Okay.
need to count, right? You
see.
don't need to do the like do like complex computation.
So do you also need to like, do you think it's also like the variable selection involved? Like,
Yes. We have 50 features in total.
Okay.
So you sort of specify how many features we want to try, right? And we also have a sort of try also different integer weight, but for simplicity, 1 is the best, right? You just count. You do need to multiply or add, right? This is, I think, one of the thinking is we want to prioritize on simplicity also.
So for this, so this is a drug approval, right?
Yes,
And
it's under EUA. It's not a
and
technical,
okay.
yeah.
And then the clinical study that was used to support this approval was a clinical trial.
Yes. Oh, so this one is a little bit different. So let me go back to here.
Mhm.
So the clinical study already done,
Okay.
and this treatment was approved in Europe because they have the SUPA.
Okay.
They have a SUPA, then they can use SUPA to guide the treatment, right? But in United States, we don't have that. We don't have SUPA. But the clinical trial was done. Now we need to find a bridging strategy. Try to determine which patient have the eligibility for this treatment, right? Then we develop this algorithm.
Okay.
Yeah. We don't want to link the efficacy outcome with this algorithm. We only want to link the SUPA level, right? Or baseline to baseline, right? We use a combination of baseline to predict a lot of baseline information.
Also, I'm wondering in general for this fact sheet of drug, so this label
Yeah.
guidance thing, it's FDA's responsibility or it's actually the so the pharmaceutical company, I think they also need to provide.
That's a very good question. So basically, the sponsor will sort of have a template, or we provide a template to them, and they will sort of finish the bowl part, right?
Yeah.
And then they give back to FDA. We will do the sort of clean up work.
Okay.
Yeah.
But like, I think they probably also done some, like, for example, if they have done this kind of analysis and then we submit.
Yes, they did.
Then you will need to like kind of like reproduce or like maybe recheck them.
That's a really good, yeah, that's a good one. So basically they provided their algorithm, but we don't believe meets the regulatory regulatory sort of standard. Then we conduct our analysis. Yeah, that's.
So, sorry, I guess I need to make sure I'm on the same page. So then you already have this study, but there is a key criteria, key eligibility criteria
Yeah.
to use in Europe is not widely available in US. That's why you try to. So then your object is actually to come up with some other criteria that hopefully allows you to replace the European the criteria SUPA.
Yes.
So in that sense, then the model that you're building is just basically purely based on
Baseline.
SUPA and
Yeah.
as well as other variables.
Yes.
And you all you used are just the study data itself.
Yes, all the
Okay.
baseline.
Okay.
Yeah.
Oh, you have same trial. as
Yeah, we have
validation.
external, and I also have additional like study, try to sort of further validate. I think one thing maybe, you may be curious is why we don't want to sort of approve this on the general population, because there are other trials did on the general population regardless of the super, it failed. So because consider the mechanism of action, right? This one is will inhibit your immune system, right? Try to reduce the cytokine, right? So if that patient they don't have any like a hyper inflammation in their body, if you use that one, that's not good. So that's why we didn't approve this treatment on a general population. It has to be very selective. That's actually refer to the enrichment strategy. Yeah.
Okay.
Yeah.
So this enrichment actually, it's interesting if I understand correctly. So in your test training and testing process, it does not really involve any outcome at all.
No.
Surely.
Yeah.
Then
We do have a exploratory analysis in terms of we link the score defined population. What is their efficacy? That's really good, actually. We can observe compared to score positive and score negative. The score positive population, they have a better mortality, like reduction. So if you're interested in that, I will show you, I will send you the paper. You can just
Sure.
check.
So then, but when your evaluation, you're essentially just using the super greater than 6
Yes.
as the outcome. and
Yes.
then okay.
Yeah. There's no efficacy endpoint involved.
And did you?
All the baseline.
Okay. Interesting.
Because the ultimate goal is to find an alternative way to identify patients that align with the super high, right? That's a baseline to baseline question. It's not
So
which
these two studies actually have both measurement.
yes,
all have
yes,
super, yeah.
yes, yes.
If you
Okay.
have the outcome, you wouldn't do this kind of
Yeah.
analysis, right? Just go ahead and use the outcome. That's even better. So
No,
right now
actually-
they're using European criteria as the sort of a
Gold standard.
pseudo outcome.
I think that's probably because it's a repurpose
Repurpose drug.
drug.
Yeah, and
Like
also-
further-
I see.
yeah.
Yeah, so that I think that's a kind of interesting, at least from my experience, is that when we typically do referring to enrichment, we are trying to enriching the patient population that's most likely benefit from the experimental regimen.
Yeah.
And when you talk about benefit, that implies that you have better outcome. But
That's a-
here,
That's a called design stage.
yeah.
Yeah, this is
no.
yeah. This is sort of like we believe this treatment is good, but we don't know how to select the patient, right? We don't have the super, but we have to find a way to define the population. So that's the sort of the
So there's already a belief that the
Yes.
treatment is good.
yes.
Yeah, that's interesting.
Oh, yeah.
Yeah.
A bit of a basic question. So
Yes.
It sounds like you're choosing people from that Save More trial.
Yes.
And people that are enrolled already have high score, right? And then
Yes.
you're supplementing, I assume, the people who have failed to enroll in the trial. Did you is there like imbalance in like the number of people that have failed to enroll versus enrolled in the training set? Like you have a lot of people from that trial and then you just have a few people. who-
That's a really good question.
Yeah.
So this one is a little bit different. Is so a lot of times when we talk about the sort of the criteria here, right? This is sort of we have a multiple additional like criteria on top of that. So first you have to be demonstrated the positive result of SARS-CoV-2, right? You need to have ammonia. There are a lot of criteria add to that.
Yeah.
So this is not the sort of the ultimate. You need to demonstrate certain other you still satisfy other certain criteria, then you can apply the rule.
Yeah.
Yeah, I know that there is an imbalance issue, potential imbalance issue.
Okay.
Yeah. Okay. Yeah, that's a really good discussion.
Oh, sorry.
Yeah.
One last question.
Yeah.
So after you apply your newly defined US criteria,
Yeah.
eligibility criteria.
Yeah.
Have you looked at the treatment effect?
Yes.
Is that comparable to the European?
We don't have any data from them.
No, but I mean, in the European, in the original European trial, you do have a treatment effect, right?
Yes, yes. Okay. I want to clarify. This trial is European trial.
That's right.
Yeah.
But
So you mean like if we check the subpopulation defined by the score, right?
yes,
Yes, it's really good.
yeah,
It's
and
good.
then is that treatment effect is comparable to
Yes.
the European trial
Yes.
that uses the original super definition.
We did the subgroup analysis.
You
It's
see
comparable.
Yeah.
Yeah.
You see what I'm asking?
Yeah. It's comparable, and also the mortality reduction is even better.
Okay.
So because our algorithm actually select a subpopulation in the super high, which turns to a severe.
More severe. Okay.
Yes.
Actually, it's a subset.
Yeah, so it's a more severe subset of the super high. So you can expect the mortality reduction will be better. So that's actually, it's more like at the early stage, you should have a clear like expectation of the tool can achieve, right? So yeah, this one is sort of a take home message. I think the importance of this is the first time we use this technique to determine a severe population for drug therapy. I guess there will be a lot of follow-up for the company, right? They can sort of analyze what are the consideration here and can they apply similar strategy in their trial. Okay, and then I guess I will go to the second one, which is also an enrichment strategy. This is a sort of a pre-specified enrichment strategy. So a drug B fails to reach the statistical significance in the phase 2 study, and they did some exploration. They found the unexpected large improvement in the primary efficacy endpoint for the placebo arm. That means in this study, they observed a strong placebo effect. So they're wondering if they can explore some machine learning algorithm to identify patients with abnormal patterns. That means before going to the randomized trial, they want to exclude the patient, exclude some patient. They want to make sure they can reduce the variability. So that's their philosophy. And the sponsor proposed to use machine learning based enrichment strategy to for 2 planned phase 3 study. So the goal is to decrease inter-subject variability prior to randomization. So they use the isolation forest. Maybe next slide we can know better. And here is what's their data source. It's very interesting. So their drug is the drug B. So they use a phase 2 trial of drug B and multiple trial of drug C for the same indication. Basically, the same MOA, right? So they can borrow some information. They hopefully the placebo cohort is very similar, right? Because for the same indication, the placebo the placebo cohort should have very similar structure.
Are you saying like the drug C is
Different one.
It's a different
Drug,
okay.
but within the same class. Yeah,
Mhm.
that's why for the same indication. So then they can sort of borrow the cohort, right? The placebo cohort, right? That makes sense, right? So they use the isolation forest. The anomaly score is calculated by applying the trained isolation forest on the components of the efficacy endpoint at the screening and the baseline. So let me explain a little bit. They don't use any post-treatment data. They use, so screening is actually earlier than baseline. So screening and a baseline, they have like 10 days difference. They sort of, and then they compare the difference, sort of how to say. So basically they compare the structure of a patient for their screening and a baseline and see if they're, how different are they, right? Because theoretically speaking, for a normal patient, at a screening stage and a baseline stage, they have only 10 days difference, right? We'll expect their sort of efficacy endpoint very similar, right? And also this one is a 10, the primary endpoint is a 10 component, is a 10 items. So you will expect, if this patient is normal, we can expect their structure should be very similar because they only have 10 days difference, right? They're supposed to be very similar. But the anomaly patient, they will have sort of like weird behaviors. Maybe their structure change, right? Their 10 items score changes. That's why we can use the screening and baseline and 10 items primary endpoint to detect the changes. And the patient with larger change will consider as anomaly subjects. So this is how the algorithm works. The anomaly score for a given subject indicates how easily this subject can be isolated from the rest of the subjects. So basically you can compute a score. So for the patient, if the score larger than a certain threshold, we believe it's anonymous subjects. We want to exclude that into a trial. And otherwise, it's a chronic subjects. We want to include the patient, and then we do the randomization, right? Then the sponsor conducted the retrospective analysis when they to the trial, the drug B, they observed the larger effect size. In that case, previously, if their p-value is 0.051, right? Then large effect size means 0.04, right? That's critical 0.01, right? So, and also similar trends were observed when they apply this to the drug C. So here is the comments we gave. They plan to use to apply the machine learning based inclusion criteria to 2 phase 3 studies. Our suggestion is we want them to use only in one of the planned study because they have potential like challenges. For example, the generatability. So it will be helpful to include the abnormal patients in at least one of the adequate and well-controlled study. Yeah, sort of like a balanced risk of the model, right? Okay, this one is another example and a very interesting one. So I guess everyone here knows the covariate adjustment, right? Right?
Yeah.
So covariate adjustment is a very is a well established one. I think a lot of the stats 101 will know that, right?
They all know that.
Yes,
Yeah.
great. So we know one critical part of the covariate adjustment is that you want to make sure the feature you include should have a certain prognostic effect on the outcome, right? In that case, you can fully leverage the covariate adjustment to reduce the sort of confidence interval or type 2 error, right? Not type 1, type 2 error. So one of the sponsor, they want to sort of use this covariate adjustment, but the way they choose the feature is a little bit different from our like convention. Our convention is we pre-specify certain variables, right? But now the sponsor says, okay, we don't want to do feature selection. We just want to build an end-to-end model. The model will predict the natural disease progression under a placebo. So basically, this one is more like they use their model to generate a new score. The score is called prognostic score. And they use that score into the covariate adjustment. That's a novel part of this summation. And they demonstrate if they there is no data drifting or covariate shifting, something like that. And they approve their algorithm or achieve the least bias, right? Am I right? So so the interesting part of this one is FDA have a well-established guidance for this type of like a covariate adjustment, right? We don't think there are any issue to inflate type 1 error, right? Because it's there for a long time. So covariate adjustment can be performed with covariates that are not prognostic, but they may not be any gain in precision compared to unadjusted analysis. So here is the framework we use. When we talk about the model risk, we have two angles. The first is the model influence, and the second one is the model risk. Usually we multiply each other to derive the final model risk. So you can see here, we know the algorithm itself is a special case of ANCOVA, right? Which have a lot of the well-established statistical properties, right? A minimum impact on the bias or the type 1 error, right? So that means if there are any issue with the algorithm, it will only inflate the type 2 error, right? So the type 2 error means it's a sponsored risk, right? They fail the trial, right? So that's why we believe this type of exploration is fine. You see, this is the comments we gave. I think the most important, the second one, the use of Percova in the primary analysis in the phase 3 trial may be acceptable. That means the model risk is low because we don't think this will inflate type 1 error. If the algorithm inflates type 2, it's a sponsor risk. And this is more detail. Because they use the Bozeman, I think the Bozeman model. Is that Bozeman? Yeah, they use a generative model, Bozeman. So that's why we wanted the sponsor to provide us a seed, because every time it might give us a different prognostic score. So we want to make sure we have everything fixed. Otherwise, they can cherry picking, right? Can repeat multiple times and give a better prognostic score, and they can do the covariate adjustment to inflate the type 1 error, right? So we want to make sure everything is deterministic with a fixed seed. Okay, this is so far for the review. Any question before I move to?
Well, just really quick, we probably have a lot more to cover. For this AI generated digital twin for Percova,
yeah,
what's the setting? Like, is it just a new way of covariate adjustment in a randomized phase 3, or something else?
You see, it's sort of like a particular application of the traditional ANCOVA, right? So so usually when we refer to the guidance, the FDA guidance for the covariate adjustment, usually we pre-specify certain important feature, right?
Yes.
But now we pre-specify the model.
Okay, so
yeah,
that's just a pre-specified algorithm for covariate adjustment
yes,
in the context of randomized phase 3.
Yes, exactly. Yes, great.
So where's the digital twin?
Yes, that's my question actually.
A lot of times, you need some gimmick, right? You need some fancy word.
But in fact, all three models, even though you talk about machine learning AI, but if I want to use the usual statistical model, that's also possible, right?
Yes,
Yeah.
yes, exactly. Yeah.
Why machine learning? I can use the logistic or linear regression. Or
so if you can solve your problem with logistic regression, everyone will be happy.
Yeah.
Right.
Yeah. Oh, yeah. Machine learning, it's kind of like just let it run.
Yeah.
But it doesn't have a confidence interval.
But we
has
do
other shortcomings.
Yeah, we do ask they provide certain uncertainty quantification if possible. Yeah, because some like a risky decision, you need to have certain like, right, your like uncertainty quantification, right, or confidence interval is not good enough, right? Confidence interval in terms of your performance metric, your outcome, you need some certain uncertainty
Yeah. So
quantification.
what's the general procedure now FDA usually use for uncertainty quantification? Because some regular techniques, for example, like dropout, people think the results is not, the uncertainty quantification is not really good, and also maybe some like bootstrap methods, but you know, it's time consuming.
Yeah.
So I'm wondering, like, in general, when you need to do the uncertainty quantification, what kind of methods do you usually use?
To be honest, I don't know. Yeah, because usually we will see, okay, you give us two different trial, you give us two different cohort, then you solve the question, right? Give us more data, more evidence in terms of you bootstrap on the same one, right? You never know the BIOS source, right? Sometimes maybe the BIOS is come from the model, sometimes from the data. You never know, right? Just give us more data. Okay, I guess next I will sort of touch base on some of our research. Actually, a lot of our research is focused on the regulatory sort of research. Is more like, can our research support FDA's mission, or at least solve some of the issue in the review, right? This is the regulatory research, because we our budget is from the taxpayer and the companies, right? So I guess I will talk the algorithm used in anakinra. Basically I design an end-to-end algorithm to sort of learn a scoring rule. I think one thing very important is we need to have that in mind, is for high stake decisions, we prefer interpretability over the performance, right? If you know some of the bad consequence of algorithm will give like a very bad consequence. For example, patient may be dead because your algorithm, your wrong recommendation. So in that case, we prefer the interpretability, right? So over the performance. That's one of the reasons we really want to dive into this region. So the scoring system, I guess, in oncology domain, there are a lot of the scoring system, right? So this is my research. Also work on the oncology. I will show you later. So previously, when we designed the scoring system, at least from my experience to read the sponsors package, and they use a stage wise, right? Basically, they have the logistic regression or have the decision random forest. They got the feature important feature, and they sort of binarize them and also come up with some weight, right? It's pretty much stage wise. It's not optimal. So in that case, we actually use a new network. We try to combine the three components together. We want to simultaneously learn the important feature and the cutoff for that feature, and also the integer weight for that feature. We want to try to wrap up as an end-to-end learning approach. This is one of the application of the ScoreNet, and we have 6 trials, and this is a solid tumor FDA data set. So it's and you can see based on the time of the trial, we hold out the latest trial, which is an external testing data set. And we use the first 5 trial as a training data. And we want to build a scoring rule to predict the prognosis of the solid tumor. And you can see the AUC is 0.7, and also we use XGBoost is still 0.7. We believe there is no big difference if we use very complicated algorithm. So that's why we use this very simple one. We have 8 features in total, and each feature was binarized, and the weight is 1, 2, 3. So it's very easy to calculate. And once you get the score, and based on the bean, so we have 3 beans. I think 0 to 10 is low risk. 10 to 19 is a moderate. Larger than 19 is a high risk. So you can see we have very good separation, and also we have 5 different treatment type. So mixed together. So you can see. So the first 3 is based on the sort of the Kaplan-Meier curve for 3 different treatment. I think some of them immunotherapy and a combination of immunotherapy and a chemotherapy. So you can see very good separation. We do trial by trial, and also in the testing data set, we have the by treatment type. Yeah. So this one is very easy to use, right? You can either use as a prognostic score for the covariate adjustment, right? And next one is the enrichment strategy. This one is the schizophrenia. So basically, a lot of the psychiatry disease, the placebo effect is strong. So basically, that's one of the reasons why psychiatry trials have the highest failure rate, because of the placebo effect. So one of the goal here is, can we identify the placebo responders? We tried, it fails. So basically it's so hard to identify the placebo responder. The AUC is less than 0.6. So instead, we don't want to identify the individual placebo responder. We want to identify, can we identify subgroup? Yeah. So the idea here is we build an algorithm. We predict the placebo responder. Of course, it's not good. In the individual level, only 0.6 AUC, but we can define those sort of subgroup as a placebo responder cohort, and we try to remove the placebo responder cohort because in the clinical trial, the treatment arm and the placebo arm is pretty balanced, right? If you remove a small population in the placebo arm, you can remove the same population on the treatment arm, right? Then you can do the sort of a post hoc analysis, right? And you can see, can we improve something if we remove some placebo responder? So this will be some placebo lead-in trial. So the philosophy of the placebo lead-in trial is you enroll several patients in the trial. You wait for 2 weeks, and you exclude those placebo responder. Then the left population will be randomized. So this one will be a good strategy for the placebo leading trial. And here you can see, so let me explain this one. So basically, the x-axis is a threshold for the x-axis is a threshold for the placebo responder. That means change from baseline. If the patient have a reduction of 10%, then the patient will be defined as a placebo responder. For example, here, if it's 20%, if the change from baseline is 20%, then we define the patient as a placebo responder. So we play with that. We see if we remove the patient with 10% change from the baseline, and we can see the sort of the gain is huge, because previously, I think you see, if we don't change anything, we have the treatment difference -5, right? But if we exclude a certain patient, the trial will be success, because you have enhanced p-value, right? The p-value will be significant. That's why this one actually is similar to reduce the variability, right? The enrichment strategy. I guess that's the end of my talk.
Right? Very interesting talk. Any